let's move on to the introduction on pharmaceutical analysis now. First, we are going to see about, look from bottom of the slide, rules and regulations on the subject. Then introduction on pharmaceutical analysis. Then overview on pharmaceutical analysis syllabus, both theory and practical. And then overview on separation techniques. Then spectroscopic analysis. Then on electrochemical methods of analysis, and about quality assurance. Let's see about overall view on your pharmaceutical analysis syllabus. Before that, introduction on pharmaceutical analysis and rules and regulations on the subject, what you need to follow when in online or offline classes. Rules and regulations for pharmaceutical analysis theory subject. First one is time management. If 11 to 12 class means you must enter the classroom before 5 minutes. Okay. And after by 12 o'clock the class will get over. So stick on to that time for both theory and practicals. This is the most important. Based on time management only, your attendance also will be marked. Okay. If you are coming late, your attendance will be considered as absent. If you are not following any other rules, then it will be reflecting on your attendance. Then you must be in uniform. And whatever rules you need to follow according to the clinical requirements, you need to follow for the uniform. Then discipline. Then brown sheet covered notebooks you must have for class, theory class, practical class, and class test. For 3 notes, for 3 purpose, you need to keep brown sheet covered notebooks. Then be attentive to the class. Then revision will be carried out for every class based on the previous class. Then assignments after each unit completion, you will be given with a group assignment. You need to submit on the given title. If you are coming regularly to the college, then it will be given with a group assignment. When you are having online classes, after completing a single unit, you will be given with individual assignments on the specific topic. Then after completing a separate chapter, you will be given with class test. If it is online, then you will be given with MCQ test. If you are coming regularly directly to the college, then you will be conducted according to your exam pattern class test after each chapter. If you have 13 units, then you will be having 13 class tests likewise. Then those who are interested, you can take seminars on interested topics in the syllabus of pharmaceutical analysis. So these are all the academic requirements for pharmaceutical analysis theory. Time management, uniform, discipline, brown sheet covered notebooks, be attentive to the class, then revision on each class, then assignments after each unit, then class test after every chapter, then seminars on interested topics to the interested persons, then attendance. Seminars mandatory. Then assignments, mostly according to your syllabus, you need to submit printed assignments, not written assignments. So prefer printed assignments like document format submission. Class test will be through online means MCQ class test. Offline mode you will be having according to university pattern. Okay, this is about theory. In rules and regulations and practicals, first is time management like theory class. If 3 hours means if the lab starts by 1:30, you should be in lab by 1:25 itself. Okay, with all the accessories. Then follow discipline. Then lab accessories, what you need to bring for pharmaceutical analysis lab, you must be in apron, then lab cloth, then glass marker for marking your glasswares, and whatever accessories you are using, you will be making many dilutions. So you need to mark with glass marker only, or else labels also you can bring. Then ballpoint pen, you cannot use ink pen. You can use ballpoint pen for practical classes, then HB pencil and scale. For since you will be doing chromatographic analysis, the chromatogram paper will be drawn with pencil using scale, everything. So these are the important accessories for pharmaceutical analysis lab. Then logbook entry before using any analytical instrument is must. Then following safety rules while handling chemicals. Okay, then handle instruments with care. You need to maintain instruments properly after using. Then brown sheet covered notebook for your observation and class test. If class test is conducted on theory in the practical class, then you need to bring brown sheet covered class test notebook. Then index maintenance in the observation book. Once the practical is completed, you must, just a minute. Hello, the class இருக்குப்பா. So after every practical, you need to maintain index for with the given experiment title and what date you are completing and how many marks you have scored based on your regular viva and performance. It will be indexed with the sign. Okay, in your observation book. Then regular viva will be asked after completing each practical. In every practical class, you will be having viva. Then record maintenance. After completing and getting sign in the rough record, you can copy it in the main record and get it signed. That is in your manual. Copy the same reading for the same experiment and get it signed. Then attendance. If you are not performing or following any rules and regulations properly, then it will be reflecting on your attendance. You will be given with absent, and you need to mind that you have to maintain 85% attendance. 82 minimum, 80 to maximum 85. Okay, 80 to 85% attendance you need to maintain. If you are coming 100% also, very good. Okay, this is about rules and regulations on pharmaceutical analysis practicals. Time management, discipline, lab accessories, what you need to bring, logbook entry, following safety rules while handling chemicals, then handling instruments with care, then brown sheet covered observation books, index maintenance after each practical with a signature, then from the staff in charge, then viva, record maintenance, then attendance. Be regular to the class. Okay. Coming to the subject. Introduction to pharmaceutical analysis. What is pharmaceutical analysis? What are pharmaceuticals? Pharmaceuticals are nothing but drug substances or drug products. Drug substances or drug products. What are drug substances? They are nothing but raw materials, active pharmaceutical ingredients, raw materials. Raw materials means as such after synthesis. What are the source for drug substance? What are the sources we can derive drug substance? Through natural source like herbal product. Okay, then through synthetic way, that is synthetic product, then to mineral source, marine source, okay, then biological source. These are all the various sources for drug substance. After deriving a drug substance, we'll be converting that drug substance to according mode of administration. So that form is called a drug product. Okay, drug product is nothing but formulated form of drug substance like tablet, capsule, injection, whatever mode we want to administer the drug to the patient. Accordingly, it will be formulated. So that is called drug product. So pharmaceuticals will comprise about drug substances or drug products. Okay. So what is analysis? Analysis means to determine identity, quality, quantity and purity. To determine identity, quality, quantity and purity of drug substances or drug products. Okay. So through analysis procedure, what you are going to determine? You will identify the drug substance, what category it is, what type of drug substance it is. Then you will determine quality of it. Okay, whether it is pure or it is related to that particular group or not. We will be checking the quality. Then quantity, how much it is present. For example, if a 100 milligram tablet is prescribed for some drug, then whether it is 100 milligram is there in that formulation or not, that we are going to determine by using analytical procedures. So quantity is determined, and we'll be checking the purity of the, if quantity is meeting 100%, then the drug is pure and it will be effective and safe for the patient to administer. So how we will check these parameters? By using analytical procedures, okay, analytical methods. So what type of analytical methods we'll be using means there are two broad categories under pharmaceutical analysis. They are quality control methods and quality assurance methods, QC and QE. So pharmaceutical analysis has two divisions, QC and QA. So what is pharmaceutical analysis? To determine identity, quality and purity of drug substances or drug products. Okay, to get this information, we have two different analytical methods, that is analytical broad analytical techniques. They are QC and QE. What is QC? Quality control. Okay, quality control is product oriented, where quality assurance is process oriented. Quality control is product oriented, quality assurance is process oriented. So pharmaceutical analysis is divided into quality control and quality assurance. Let's see about what are quality control methods. They are product oriented. That means it is related to the product which we analyze. Okay, product oriented. This is process oriented. So whatever methods or procedures we follow for analyzing these drug products are called as process oriented. That we need to follow all quality based procedures. Okay, so that is monitored by quality assurance procedures, and quality assurance will give about all standard guidelines which need to adopt for pharmaceutical analytical methods, that is quality control methods. Where quality control product oriented analysis is divided into two categories, qualitative analysis and quantitative analysis. When you carry out qualitative analysis for a drug substance or drug product, you can identify you can find this parameter identity you can get from qualitative analysis. Qualitative analysis is nothing but just a deduction method, whether the compound is related. For example, if the compound is having some basic nucleus of what, ma? Somebody wants to take. Whether clear?
Ma'am, Jacqueline, ma'am.
Uh-huh.
Ma'am, Jacqueline wants to join the class.
Oh, join now. Okay, just wait. Okay, everybody has joined, no? Mute yourself. Quality control is product oriented, where it has two divisions, qualitative analysis and quantitative analysis. Qualitative analysis will tell about means, by means of that we can identify a compound which class it is belonging to. Okay, whether it is belonging to some chemical group. If you get some basic nucleus like amino acid or protein, whatever you carbohydrate or whatever class it is, we can identify through some chemical tests. Most probably will be using identification tests under qualitative analysis to detect drug substances. It is basic methods, qualitative methods. And even by using instrumental methods, we can analyze qualitatively drug substances and drug products. We can identify using many parameters. So who is that? Mute your mic. And how this analysis is carried out means based on the physical and chemical properties of drug substance. Okay. On what basis we will analyze its identity, quality, quantity, and purity means this analysis is carried out based on whether you are following, all of you are following or not. Hmm? So how we are carrying out analysis means based on physical and chemical properties of drug substance. Okay, how mainly physical and chemical parameters are followed to determine its identity, quality, quantity, and purity. So both by qualitative and quantitative analysis, we'll be seeing physical and chemical properties, whether the drug is solid, liquid, or gas. Likewise, physical properties, and like chemical properties, whether it is belonging to some acid category or alcohol category, amines or aromatic, aliphatic, what type of chemical nature it has. Accordingly, taking that into consideration, we'll be analyzing drug substances or drug products by qualitative and quantitative analysis. And then in this qualitative and quantitative analysis will be studied by there using means by means of two broad courses subjects. First, in the first year you would have studied PIC, right, pharmaceutical inorganic chemistry, where you should have followed volumetric analysis like titrimetric analysis. And these are all the basic pharmaceutical analytical methods to quantify and to check the purity of drug substance. You should have done assays, right, to determine percentage purity. So that particular experiment tells about the determination of purity of drug substance, okay, through volumetric analysis. Volumetric analysis is very basic methods. And now we are moving to pharmaceutical analytical methods. They are nothing but instrumental analysis, using instruments with a minimum quantity. What is the advantage means comparing with volumetric and instrumental analysis? In volumetric analysis, you will be using more amount of sample. At least in gram you need to take for analyzing by volumetric method. Like at least 1 gram, 2 gram sample you need to take. For instrumental analysis, you require from microgram to milligram amount only for analysis. So with that microgram to milligram amount, using various analytical, sophisticated and preliminary analytical instruments, you are going to analyze drug substance or drug products. So whatever you want to determine its quantity, purity, whatever you need to analyze, any category you need to analyze, then you will be using that particular instrument. So this is the category you are going to study under pharmaceutical analysis. Okay, there are various types of instruments. Like instruments are divided into spectroscopic and chromatographic instruments. By means of those instruments, you are going to determine determine the quantity of, or you are going to carry out both qualitative and quantitative analysis for the drug substance. Selected analyte, we can say the sample as analyte here in the analytical methods when you are carrying out. Okay, when you are carrying out analysis, it will be called as analyte. Drug substances or drug product may be called as analyte. So that analyte, according to its physical and chemical properties, will be choosing the particular instrument which is relevant to that category of sample, and will be analyzing it qualitatively and quantitatively. So we are going to discuss about various analytical methods under various instrumental methods under pharmaceutical analysis. Okay. So hope you have understood the difference between volumetric analysis and instrumental analysis. Volumetric analysis are basic quality control methods for drug products, where instrumental analysis is modern or sophisticated methods for very sensitive methods also for analyzing your analytes. Where quality assurance is process oriented, it will ensure the safety and efficacy, we need to follow all quality control and quality assurance procedures. So you must learn what are quality control methods available for analyzing the drug substances, and what are the quality assurance guidelines we have for carrying out all quality control methods to achieve safety and efficacy. This is the main aim and objective to study the pharmaceutical analysis subject. Even clinical samples, after administering drug to the patient, if you want to determine the bioavailability, and then you need to carry out quality control methods using analytical instruments only. How it is carried out? By using analytical instruments, and to ensure the safety and efficacy by means of determining its purity. Okay, so that only the particular subject is very important in Ph.D. course. Okay, so you must learn instrumental analysis clearly. So you need to know the principle, how to operate instrument, and what type of samples are analyzed, and how much sample we need to prepare, what are the preparation procedures, what are the deduction procedures, how you will analyze the results. Everything you need to you need to study, that you are going to study in the pharmaceutical analysis subjects. Next, let's move on to what are the, according to your syllabus, what are the different category of topics available. Okay, you're dividing into, your whole syllabus is divided into 4 divisions. So before moving on to the syllabus, let's revise from beginning. So you are going to see about rules and regulations, pharmaceutical analysis introduction, syllabus overview. Then under syllabus there are 4 categories, their overview. Okay, separation techniques, spectroscopic analysis, electrochemical methods of analysis, quality assurance. About theory, rules and regulations, these are all the parameters you need to follow. Main thing, assignments, revision assignments, class tests, seminars, these are all the works will be given after each and every class. You need to maintain class book and attendance, time management, everything. Then for practicals, accessories, what you need to bring, logbook entry, important rules and regulations you need to follow, index maintenance, viva, record maintenance, and attendance. Then we have discussed regarding what is the importance of studying pharmaceutical analysis. What are drug substances, drug products, what is the use of analysis, and what is meant by QC and QA methods. QC is nothing but product oriented, it is qualitative and quantitative analysis. It comprises of quantitative and qualitative and quantitative analysis. Then subjects covers these analyses, PIC and VA for Ph.D. PIC you should have learned all about volumetric analysis, where you should have, these are the basic analytical methods to determine quality and purity. And pharmaceutical analysis will tell about all instrumental methods of analysis for analyzing your drug substances very sensitively with specific instrumental techniques. Then quality assurance is process oriented. Whatever procedures you are following here for QC method is followed based on the guidelines given under quality assurance to ensure safety and efficacy. Everything is carried out to ensure safety and efficacy of the drug product, so that we can give confidently to the patient for the treatment. Okay. Now let's move on to the syllabus overview. So syllabus is divided into 4 classifications. Okay, 4 categories: separation techniques, This is the instrument used for separation technique. Okay, sophisticated instrument called HPLC, separation techniques. Then spectroscopic analysis. We are going to study about spectroscopic analysis. So four broad categories we are going to discuss. One is separation techniques, spectroscopic analysis, then electrochemical methods of analysis by means of electrodes. You should have seen pH meter. So analysis by electrodes attached instruments. Okay, if you analyze your samples, then it is coming under electrochemical methods of using electrodes. Chemicals are analyzed. So what are the different methods we are using, and for that is coming under electrochemical methods of analysis. Then quality assurance. So this three part will cover QC, quality control methods. The fourth part is quality assurance. Okay, separation techniques are carried out by using the instrument called chromatograph. 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 Instrument name is chromatograph. So chromatographic techniques are nothing but separation techniques. Why it is known as separation techniques? Will be separating mixture of drug substances into individual molecules by separation techniques. Okay. And next about spectroscopic analysis using different spectrophotometers. This is UV visible spectrophotometer which is available in our campus. So spectroscopic analysis using why the name derived as spectroscopy means we'll be using radiation for analyzing your sample, and by means of applying radiation we can determine the characteristic of drug molecule, what chemical structure it has. We can structural characterization we can done with spectroscopic analysis, where we can separate the molecules into individual or pure molecule by separation technique. So combinedly both the techniques will give complete information about a drug molecule. Okay, separation technique and spectroscopic, these two are very most important techniques under pharmaceutical analysis. Chromatographic techniques and spectroscopic techniques. Chromatographic techniques are nothing but separation techniques. Spectroscopic techniques are nothing but using radiation samples are analyzed. What type of radiation means? Electromagnetic radiation. This we'll discuss briefly under the particular specific topic when we are getting into. Okay. Then electrochemical methods of analysis by means of using electrodes. There are various types of chemicals are analyzed according to the nature of chemical. We'll be preferring different types of electrochemical techniques. So whatever we are going to study under this are called as electrochemical methods of analysis. Then these are covering quality control methods under pharmaceutical analysis. Then quality assurance guidelines. To perform these quality control methods, what are the standard guidelines are given under quality assurance to get 100% quality of the drug product, okay, before administering to the patient. So the things that covers under pharmaceutical analysis we'll be discussing. Okay, so these are all the four broad categories. Now let's see the overview on separation techniques. Shall we discuss this part in the next class or will cover now itself? Give me a suggestion. If you are ready, I will cover the whole PPT, or else we'll discuss in the next class. If you get bored, then we'll discuss in the next class. Okay, anybody answer?
Your wish, ma'am.
My wish. Okay, so you all are able to listen, no? I'll take slowly then.
Yes, ma'am. You can take.
Okay, thank you. So we'll complete about all overviews on your complete syllabus, what you are going to learn. So now let's move on to the separation techniques. What are the different separation techniques you are going to study? Separation techniques, what I told, they are nothing but chromatographic analysis. We are by means of chromatography, we are going to separate. You can see the picture here. We are separating the molecules into individual molecules on the chromatogram. So for this separation from mixture is called as chromatography analysis, so that they are called as separation techniques. There are different separation techniques are available, and then this separation techniques are again classified into planar chromatographic analysis and columnar chromatographic analysis. Okay, separation techniques are classified into main two categories: planar techniques and columnar techniques. Planar techniques means the separation occurs over the plane surface. See here, plane surface. It is HPTLC. You can here also like similar to this plate separation, plane surface separation occurs. Okay, so that is called planar chromatographic analysis. What are the different types of planar means? Paper chromatography, thin layer chromatography. TLC is nothing but thin layer. You can see the thin layer. Over the thin layer, sample is getting separated into individual components. So this is called thin layer chromatography. This is high performance thin layer chromatography, which is applicable for quantitative analysis. Mainly quantitative analysis is carried out by HPTLC, where TLC and paper are qualitative and semi-quantitative analysis. Okay, we can carry out qualitative and semi-quantitative analysis by paper and TLC, where by HPTLC we can go for a sensitive quantitative analysis for the separated molecule. Okay. So this is about planar chromatographic analysis. What is the other part of separation technique? Columnar chromatographic analysis. In the column, either open column or closed column. This is open column, this is closed column. We will be separating our drug products or drug substances. What are the different types of columnar chromatography means? Hence it is called as columnar chromatography. Since we use columns, either open column or closed column, it is called as columnar chromatographic analysis. It is highly sensitive when comparing to planar chromatographic analysis. Okay, when comparing to paper and TLC, this columnar chromatography is highly sensitive since it is separated through the closed column. Okay. And what are the types available means? Column adsorption chromatography. By means of adsorption procedure, in the column separation occurs. So it is called as column adsorption. Performance liquid chromatography. Then ion exchange chromatography. It is a modified form of HPLC only. In the column exchanges, ion exchanges are used for ion exchange purpose. Okay, thereby the samples are separated. Then GLC, gas liquid chromatography, is GLC instrument, gas liquid chromatography. Gaseous samples are separated by using gas liquid chromatography. So these are different types of column chromatography that you are going to learn according to your syllabus. What are the techniques? Column adsorption, HPLC, ion exchange, and gas liquid chromatography, GLC. Ion exchange is modified form of HPLC. In the column itself, we can use ion exchange column for separating the molecules. Okay. In GLC, gaseous molecules, we can call HPLC as liquid chromatography, GLC as gas chromatography. And through column adsorption, open column, it is called as open column chromatography, where we can isolate the samples from bulk amount of mixtures like herbal samples. So column adsorption is mostly applicable for herbal samples, where we can isolate individual molecules from multiple molecules, multiple mixtures, herbal mixtures. We can separate, extract, we can separate individual molecules by column adsorption. So there are three main category under columnar: column adsorption, liquid chromatography, and gas chromatography. Okay, here column adsorption, HPLC, ion exchange, and gas liquid chromatography. Clear? So what are the two broad categories of separation techniques? Planar chromatography and columnar chromatography. In planar chromatography, separation occurs over plane surface. So what are the types? Paper, TLC, and HPTLC. For columnar, column adsorption, HPLC, ion exchange, and GLC. Okay. Next, miscellaneous chromatographic techniques. Apart from that, those are major chromatographic techniques applicable for qualitative and quantitative analysis. These all will be applicable for quantitative analysis. Okay. Here for quantitative, mainly will be going with liquid chromatography and gas chromatography. After isolation by column adsorption, will be carrying with HPLC or GLC for quantification, or we can go with spectroscopic analysis for quantification purpose. Okay, or we can use HPTLC for quantification after isolation. This column adsorption is mainly used for isolation, and quantification purpose we can prefer either HPTLC according to the sample nature. We can prefer HPTLC, HPLC, or GLC. Okay. Then other miscellaneous techniques are electrophoresis. By applying electricity, the mixtures are separated into individual molecules between electrodes. So by means of electrical migration, separation occurs. So that is called electrophoresis. Phoresis means migration. So electrical migration. Through electrical migration, sample. So this is a type of separation technique, electrophoresis or electrochromatography it is called. Then gel filtration or affinity chromatography. It is also modified form of HPLC. In the closed column, it will be packed with a gel according to the separation, according to the size of the molecular size, it will be separated. So gel filtration and affinity chromatography are closely related chromatographic techniques. By using gel, the separation occurs based on affinity. Okay, both are related only, so it is so that only it is given with the same heading. Okay, so gel filtration and affinity chromatography. So these are all the miscellaneous chromatographic analysis. So what are the techniques? Planar, columnar, miscellaneous. Under planar, paper, PLC, HPTLC. Columnar, column adsorption, liquid chromatography, and gas chromatography. Under liquid chromatography, HPLC, ion exchange. Under gas chromatography, gas liquid chromatography. And miscellaneous electrophoresis, electrical migration, or electrochromatography. And another one is gel filtration or affinity chromatography. It is also similar to HPLC. So we can modify this column with gel filtration, gel column, according to the separation by gel filtration or affinity chromatography. So this is about overview on separation techniques. Overview on separation techniques. Next, about spectroscopic analysis. We'll discuss this in the next class only. I'm exhausted to explain. Then I may not say many points. So we'll discuss about overview of spectroscopy, electrochemical methods, and quality assurance part in the next class. So with this, we'll complete today. So what are the things we have discussed today? What are the titles we have discussed? About rules and regulations, what you need to follow for theory and practicals. Then on introduction on pharmaceutical analysis, about QC and QA methods, and about your course of study. That's the PIC and PA. Then on PA syllabus, in that we have discussed, it is divided into the 4 parts: separation part, spectroscopic, then electrochemical, then QA. QC is divided into 3 parts: separation technique, spectroscopic analysis, and electrochemical methods. And last one is QA is covering all guidelines. In that only we have discussed the first part, separation technique. In the separation technique, there are 3 categories: planar, columnar, and miscellaneous. In planar, paper chromatography, PLC, HPTLC, PC, TLC, HPTLC. Columnar, column adsorption, HPLC, ion exchange, and GLC. In miscellaneous, electrophoresis, gel filtration, and affinity chromatography. Okay, remaining we'll discuss in the next class. Thank you all for patient listening. If you have any doubt,
Thank you, ma'am.
ask me. Thank you. If you have any doubt, you can ask. When we are discussing briefly about each technique, you will understand better. Okay. Now I have shown you the instrument and different types, so that you will understand, you will get some idea what you are going to learn. Okay. And by using these instruments only, you are going to analyze your biological samples. When you collect blood samples or urine samples, you should have seen some clinical laboratory. If you have gone, you should have seen the instruments, instrumental analysis. They would have kept many instruments. They are nothing but analytical instruments only to analyze your clinical samples qualitatively and quantitatively. Okay. All the test, whatever you are doing in the laboratory, clinical laboratory, by means of instruments only, so that we need to learn how this instrument is operated, what is the mechanism behind operating this instrument, what are the various applications apart from clinical application. This is the main for you. Apart from it, what other samples also can be carried out, so that it will be useful when you are getting into clinical research. Okay. So that only you are going to study this subject, and it is most important subject like your therapeutics and other subjects. Okay, that's why it is kept in third year. Because after learning your basics, you are getting into instrumental analysis. Okay, thank you all. If you have any doubt, you can
Thank
ask.
you, ma'am.
Thanks.
Thank you, ma'am.
Clear?
Thank you, ma'am.
Whether I am going fast or okay?
Okay.
ஃபாஸ்ட்டாக போனால் சொல்லுங்கள், இன்னும் கிளியராக எக்ஸ்பிளைன் பண்ணுறேன். எனக்கு தெரியாது இல்லையா நீங்கள் எப்படி ரெஸ்பாண்ட் பண்ணுறீங்கன்னா, அதனால நான் கொஞ்சம் ஃபாஸ்ட்டாக போயிருக்கலாம். இல்லை டைரெக்டாக வந்தப்போ உங்களுக்கு இன்னும் ஏதாவது டவுட் இருந்தாலும் கிளியர் பண்ணிக்கலாம். ஓகேவா? இந்த மெட்டீரியல் ஐ பி அப்லோடிங் இன் யுவர் கிளாஸ் ரூம்.
Okay, ma'am.
ஓகேப்பா.
Okay, ma'am.
தேங்க்யூ.
Thank you, ma'am.
Thank you,
Thank you,
ma'am.
ma'am.
தேங்க்யூ ஆல். தேங்க்யூ. மேக்ஸிமம் எல்லாரும் ஜாயின் பண்ணிருங்க. ஓகேவா? ஒரு 5 மினிட்ஸ் முன்னாடி நான் இல்ல கரெக்ட் டைம்க்கு மீட்டிங் லிங்க் கொடுத்துருவேன். இல்லை 5 மினிட்ஸ் முன்னாடி அப்லோட் பண்ணுவேன். இப்போ இமிடியேட்டா ஜாயின் பண்ணிட்டீங்கன்னா ஈஸியா இருக்கும். கிளாஸ் ஸ்டார்ட் பண்ணா வீணை அட்மிட் பண்றதுக்கு கொஞ்சம் கஷ்டமா இருக்கு. அதனால ஏன்னா ஜெட்லின் லேட்டா தானே ஜாயின் பண்ணியிருக்காங்க.
வரல மேம்.
பரவாயில்ல. இந்த மாதிரி இன்ஃபார்ம் பண்ணீங்கனாலும் ஓகே. மிடில் இன்ஃபார்ம் பண்ணா கூட நம்ம ஜாயின் பண்ணிக்கலாம்.
ஓகே மேம்.
ஓகேப்பா. தேங்க்யூ ஆல்.
Thank you, ma'am.